Good morning, everyone. How are we doing today?
Good morning. Your webcam's upside down.
The webcam is upside down.
Yeah, it looks like you're coming from the top.
Oh, yes, that is odd, isn't it? Let me see. Okay, now let me not waste time. I think you would be more concerned with my voice. As long as you can hear me, I'm not going to waste time fiddling around with the video settings. So I'm going to get started right away. So you do hear me okay, and that is good. Do you have any questions before we begin? Any comments to make? We're all good? Good health? Very good. So today we are covering chapter 13 in the book, which will take us through a brief trip through the field of viruses, virology. We reviewed, of course, at the beginning, microbial structure in general that was focused on bacteria, essentially. And here we will talk about viruses, which are, of course, an integrated component of infectious diseases. Right. So a couple of slides on the brief history of virology. So this is just like bacterial work. In fact, it was really late 19th century that research came to life and continued since then. There are a few names, key researchers in this field, specifically Avnovsky, Martinez, Bierning, who first determined the cause of disease due to filterable viruses. Of course, the term virus is derived from Latin for poison. So those entities are essentially classified, as mentioned here, filterable entities. Now, the discovery of key component of these entities, those that particularly attack bacteria, discovered by Twart and Dierl, what we call bacteriophages, which we will also talk about in this chapter. So those are viruses that were later discovered to attack a specific bacteria. So the term filterable viruses is actually from, I added a couple of slides. Where are the pointer options? Here, let me take you to this slide before I go back. So this is, you've probably seen this in some textbooks. This is the so-called Pasture Chamberlain filter. And this is how they used to try and sterilize liquids back then. So this would be the container containing the material to be filter sterilized. And the actual filter is this cylindrical structure it's typically metallic, containing inside a porcelain filter. And from there, material goes through the filter, comes out, of course, filter sterilized, and then it is used, it's drawn out through the action of a syringe. And that will produce material that's supposed to be sterile. But what they discovered, actually, I have another slide to show you the inside of this filter, the porcelain filter. So this is the actual porcelain filter housed within this metal cylindrical valve. And the porcelain filter is manufactured such that it contains very, very small holes that allows liquid to go through, but not bacteria. So the holes are too small for bacteria to go through. But what they discovered essentially was that this material that comes out of the filter, in some cases, it contains material that can still cause disease. The so-called, this material, the so-called poison, dubbed viruses, was able to cause disease. And that was really the beginning of the field of virology. Now, viruses have, at the time, they were also discovered to have certain characteristics that resemble more complex chemicals rather than actual cells, such as bacteria. So, for example, you can take certain viruses, not all of them. These particularly apply to the naked viruses. Some of these viruses, naked viruses, you can precipitate from solutions of ethyl alcohol or ethanol, or you can even crystallize them in a test tube. So using various experimental techniques, essentially high concentrations of these viruses, and then slow removal of the solvent, and that will cause them to either precipitate as powder or crystallize. Of course, these properties at the time were typically used to describe complex chemicals, and therefore viruses back then were then thought of being actual complex chemicals rather than actual entities. Now, viruses are acellular, as you know, because they do not resemble cells, right? They do not have any metabolic activity that's happening inside the actual virus, or ribosomes, of course, to make proteins. They're very simple. They contain the genome, the genetic material, which could be either DNA or RNA. So unlike the rest of the microbes, which only have DNA as a genome. So outside the host, they are inert. Okay. So some scientists argue against using things like viruses are dead outside the host and then become or come back to life after infection. So the term inert particles was then used instead. So again, they lack any of the properties that we recognize with cells. Again, metabolic activity, the ability to divide or replicate their genome independently of the host, their ability to move. Of course, viruses do not have any of these features. So the viruses are able to multiply only after they infect the cell. So they're obligate intracellular parasites, if you will. And then they use the cell's own replication machinery and protein synthesis machinery to replicate themselves before escaping the cell and then infecting other cells in the immediate vicinity or in other areas. So they would be released to the environment, of course, and they would spread and infect other hosts. So that is really their life cycle. they are referred to as infectious agents rather than organisms for the reasons that we've just described. The virus requires a specific host. It cannot be grown on its own in the laboratory as we do, for example, for bacteria. So we cannot use liquid media. We cannot use, even complex liquid media cannot be used, or solid media, such as agar. You have to have a host. And we'll talk about the choices of hosts that can be used to cultivate viruses in the laboratory. And as I mentioned in the last class, they are typically smaller. The vast majority of them are much smaller than bacteria, with the exceptions of a couple of species that are in fact comparable in size to some of the smallest bacteria. So those exceptions are visible with the light microscope. And I'll talk about some examples later on. In terms of classification, because of their simplicity, the classification system used to relate viruses to each other is also simple, compared, of course, to bacterial classification systems. So they can be classified according to their host, whether the host is a eukaryote or a prokaryote. They can also be classified from there according to their outer surface. Are they naked versus enveloped? Do they have RNA or DNA genome, etc. In terms of bacteria viruses that are called bacteriophages, or phages for short, they are used quite extensively by molecular microbiologists who are interested in the study of microbial and viral genetics. For the simple reason, of course, viruses multiply quite rapidly and they produce large numbers. And their genomes are small, so they can be manipulated relatively easily compared to bacteria or, of course, eukaryotes. So they do provide a good model for how animal viruses interact with their hosts, except, of course, here we're talking about a virus interacting with the bacteria rather than an animal cell or a whole animal or a plant. Viruses are a very important vehicle for horizontal gene transfer. Okay? So, of course, you know the distinction between vertical gene transfer, which is essentially the transfer of genetic characteristics from parent to offspring. And then there's the horizontal gene transfer, where genetic material can be changed or transferred, not from one generation to another, but within the same generation. Of course, bacteriophages' presence on Earth has very important consequences ecologically and medically. Ecologically, of course, because they keep bacterial numbers in check as they infect and multiply. In the medical area, they are being investigated to actually attack bacterial infections in cases where the treatment with standard antibiotics, for example, fails. Bacteriophages can be exploited to do the job because of two important features of their activity. One is their selectivity. So bacteriophages, just like the majority of animal and plant viruses, bacteriophages will attack a single type of bacterium, not others. So a phage that attacks E. coli, for example, is not going to attack any other bacteria. This is the one feature. The second feature is the fact that when the phage is multiplying, before it is released, it actually kills the bacteria. Hence, its potential utilization to treat bacterial infections. As I mentioned, some of the larger viruses, like the Mimi viruses, as you can see here, there are approximately 800 nanometers or 0.8 microns. It's relatively large, comparable in size to the smaller sort of coccoid bacteria, such as Staphylococcus or Streptococci. And this is the comparable sort of size of the bacteriophage compared to E. coli. So essentially, the typical size of bacteriophage, this particular example is called the T4 bacteriophage. When it multiplies inside the E. coli, it will produce approximately 200 bacteriophages. That's how many it can fit. Then you have, of course, one of the first viruses to be characterized, the tobacco mosaic virus. Again, so this is around the lower limit, 0.25 microns, about the lower limit of resolution for the light microscope that we typically use in the labs. And then from there, you have very, very small viruses. Here we have the poliovirus at 30 nanometers, quite tiny, or 0.03 microns. And there are actually viruses that are even smaller than the poliovirus. Somebody have their microphone on? Please check your microphone. I can hear some background sound there. Yes, Chris.
I was wondering, what theoretically would be the smallest that the virus could be that could no longer affect the host cell for human?
The smallest virus?
Yeah, like where the limit where it's too small that it would no longer be able to do damage to really infect a human.
Well, it's not the size of the particle per se, Chris. It's really whether or not the genome of the virus contains all the necessary genes for it to multiply inside the host. That's the criteria. Now,
So
in terms of the size of the genome, which would reflect in a way the size of the virus. The smallest functional virus that we know of, they're around the size of 10 nanometers. Okay.
So theoretically, could like a 1 nanometer virus still cause damage? No.
No. I don't believe, or there aren't any viruses that I know of that are that small in size. I think for the simple reason that it will not be able to fit the genetic material, the minimal genetic material that's needed for the virus to actually multiply inside the host.
Okay, so the limit factor is the genetic material is going to be too large, so it can't be any smaller than like 10 nanometers.
Yeah, exactly. That's the smallest size that we have discovered so far that is able to actually infect successfully infect its host.
Thank you very much.
You're welcome. Okay. All right. So let's go through. Oh, I went through too fast here. Okay. So let's go ahead and look at in more detail. So as I mentioned earlier, viruses are typically of two types: either the naked virus, shown here on the top right, and we have the enveloped viruses. The non-enveloped viruses is quite simple. It contains a protein coat that we call capsid. The individual protein components that make up the capsids are referred to as capsomers, and these essentially act as a shell to contain the genetic material of the virus. Now, from this simplistic view, additional structures may emanate. For example, you could have spikes that extend outwards from the surface of the capsid. The inside of the virus, in addition to the genetic material, it may contain one or more enzymes. So these are proteins that the virus requires for successful multiplication inside the host. So it carries these enzymes with it after it leaves the cell, and it will use it as soon as it infects a new cell. For the enveloped virus, it's essentially the same level of complexity as the non-enveloped virus, with the addition of an envelope. The envelope is an actual cellular membrane that it takes with it after it leaves the infected cell. And we will look at details of how this takes place. And in between the capsid and this envelope, it can also carry additional proteins or enzymes that are referred to as matrix proteins. That is essentially the structures for both. And when we talked about in the previous chapter about controlling microbial growth, we talked about the relative resistance of different forms of microorganisms. And typically, the naked viruses rank up there along with other resistant bacteria in terms of disinfectants. And of course, they have their own resistance to antibiotics, the standard antibiotics that we use to treat bacterial infections. And I'll also touch on that later on in this chapter. In terms of the shape of the capsid itself, these are the three most common forms. We have the icosahedral shape, shown here on the right panel, the top right panel. The black and white image in each of these represents an actual electron micrograph image of a representative virus. So for the icosahedral one, the image is that of an adenovirus. And the icosahedral, it's a very actually neat geometry, and it represents 20 flat triangles that self-assemble to give this icosahedral geometry. Next, we have the helical geometry, and the best example of this is the tobacco mosaic virus, which is relatively long, about 0.25 microns. The actual capsid is helical, as you can see. And inside this helical capsid, you will have the actual genome of the virus organized essentially in a helical arrangement as well. And lastly, we have the so-called complex viruses. So anything with geometry other than the first two is included in the complex viruses. So here we have the example, the T4 bacteriophage that attacks E. coli. It has the typical geometric nucleocapsid, but in addition, it has a tail, it has tail spikes, and it has tail fibers. And so here the genome is within, of course, the nucleocapsid, which is the nucleome and the nucleoid and the capsid together are referred to as nucleocapsid. So the scientific body that is concerned with viral taxonomy is called the International Committee on Taxonomy of Viruses. And it keeps essentially various databases, including published features and research on viruses, their classification, and the nomenclature. In 2020, the report from the ICTV came out describing 6,590 viral species that were classified into 1,421 different genera. So you could see the number of viral infections, although quite significant medically for us and for the animals and the plants. They represent a very small fraction of all the viral species that we have identified so far. In addition to this kind of information, the report contains characteristics on genome structure and the host range of the viruses. And it could also include things like viral shape, disease symptoms, and other features that are relevant for researchers and the medical establishment. So there's a new taxonomic system, however, and it follows the standard taxonomic hierarchies that we use for bacteria and other biological systems. So there are essentially 8 principal ranks from species through realm. Realm is the class or the group that is analogous to the domain. So we have, of course, 3 domains of life, right? We have eukaryotes, bacteria, and we have archaea. And this would be a 4th realm for viruses. Families of viruses typically end with the suffix viridae. For example, Rhabdoviridae. There is no consistent convention in terms of using the names. So, for example, we know for bacterial names, the species and the genus are written in italics. In the older days, they used to underline them, but now with the ability to use italics, they are always italicized in research reports and any scientific documents, in fact. But sometimes they're also referred to by their species name or by some other name. Okay, so here, for example, we have the rhabdovirus, could be written exactly as rhabdovirus without italicization or capitalization. Groups of unrelated viruses can also be used, or their names, or referred to together because they share the route of infection. So, for example, we can talk about respiratory viruses collectively. Those viruses that are transmitted through the oral fecal route, we would refer to as enteric viruses. Those that are transmitted from animals to humans, we refer to as zoonotic viruses. Those by arthropod vectors, transmitted through arthropod vectors, are referred to as arboviruses, right? These can, arthropods are in fact crucial vectors for the spread of a large number of viruses, and they cause some important human diseases. The yellow fever, dengue fever, West Nile encephalitis, La Crosse encephalitis, and many others that we will touch on throughout the course. So here we have some grouping of human viruses based on the route of transmission, and more examples in the right column. Okay. The route of transmission, or mechanism of transmission, as you can see, we have fecal to oral, respiratory, we have sexual contact, we have the vector, as we just mentioned, and we have direct animal to humans, such as the rabies and the cowpox. Right. So here is actually a nice electron macrograph image of a Mimi virus. Remember, in a few slides back, I talked about some of the larger viruses. Right? The Mimi virus is a mega, belongs to Megaviridae, and mega, of course, simply reflects the large size of these viruses, so collectively referred to as Megaviridae. Approximately 0.8 microns, fairly large, and that reflects. Back to your question, Chris. So the large size of this virus reflects the large size of the genome. So, for example, Mimi virus genome contains 1.2 million base pairs. That's quite large. Consider, for example, E. coli. The genome of E. coli contains approximately 4.1 million base pairs. So this is fairly large. This, in fact, is so large that itself is preyed upon by other viruses. Imagine that. There is, in fact, one virus that attacks the Mimi virus called Sputnik. The panda virus is also one of the large ones. It is so large that its genome is, in fact, twice the size of the Mimi virus. So here we're getting very close, in fact, to half of the genome of a bacterium like E. coli. Mind you, though, some bacterial genomes contains less than 500,000 base pairs, such as mycoplasmas and related bacteria pathogens. Okay, so let's go back and look at the different viruses in some more detail, and look at their life cycle. So we'll start with bacteriophages before we move on to animal and plant viruses. Bacteriophages, based on their life cycle and how they infect the bacteria, can be classified into one of three different in case anyone thank you, Alan, for sharing this. That's a very interesting virus. Now, remember though, with this Sputnik situation, this virus does not prey on the Mimi virus outside the host. Again, because the Mimi virus itself, being inert itself, it lacks the machinery it needs to actually multiply or replicate the genome. It also lacks the machinery to synthesize the proteins that are required to assemble the capsid. So the Sputnik does not actually attack the Mimi virus outside the host and multiply. To the contrary, it will only prey upon the virus if it happens to co-infect the same cell, the same animal cell. Then, during the process of replication, it will actually prey upon the system of the virus itself and multiply. Okay, so its multiplication is at the expense of the virus. Right, back to bacteriophages and their life cycle. So again, depending on how the virus or the phage multiplies, it can be classified as a lytic or virulent phage. It can be a temperate phage, where it does not actually multiply and kill the bacteria. There is also the so-called filamentous phages, which are quite distinct in that viruses typically, after multiplication, as I mentioned on several occasions, after multiplication, they are released from the bacterium by simply lysing the cell. Right? This for filamentous phages, this is not in fact the case. The general strategies for phage replications are one of two: either a productive infection, where the virus simply multiplies soon after infecting the bacteria, or it can be a latent state. And I think you're familiar with this term. So this is where the viral genome does not actually go into the productive cycle, but remains silent within the cell. The genome, obviously, of the phage at this point is replicated as the bacteria also divides. So here we have the two forms. We have the productive infection, where the release of the virus, of course, causes cell lysis and cell death. There is the release of the virus from the host cell, where the bacteria actually does not die. Okay, so it continues the release of the virions, as in the case of the filamentous bacteriophage, and as in the case of animals, animal viruses, okay, as well as plant viruses. Where we have then the latent state, where the virus actually is not active, it does not multiply, is not released, remains silent, and its genome can either be integrated within the chromosome of the host or remain as an independent piece of DNA known as a plasmid. And this plasmid, of course, is copied as the bacteria divides. So here we have the virus is silently being actually replicated and increased in number without itself actually being complete. There are 5 distinct stages to viral infections, regardless whether we're talking about bacteriophage, an animal virus, or a plant virus. Okay? 5 distinct stages. And do I have an image here, a larger image? No. Okay, refer to the images here to the right on the slide. The first stage is the attachment, of course, sometimes referred to as adsorption. Okay, this is where the virus actually, the surface of the virus recognizes a specific receptor or receptors. Sometimes the receptor itself is actually not a single transmembrane protein, but 2 different transmembrane proteins or 2 different receptors. So after the binding, and this is why viral infections are specific to host. I mentioned about the bacteriophage, the T for bacteriophage specific to E. coli, because it needs to bind a specific molecule on the surface of the E. coli. And this is true for animal viruses, okay, not plant. I'll come to that later on, but for animal viruses. So a cell that does not have that specific surface molecule will not allow the virus to absorb to its surface. Okay, normally would be considered resistant. After the adsorption step, of course, the virus needs to be able to deliver its genome to the host cell. In the case of the bacteriophage, the actual phage remains attached to the surface, and it will only insert its genome to be replicated inside and to manufacture the viral progeny. Right? So genome entry. The third step is the synthesis, where obviously the phage needs to synthesize, replicate its genome, because each viral particle to be produced needs a copy of the genome, and also needs to have the protein capsids to actually package the genome. And then fourth stage, we have the assembly. All these components, the replicated genome, the capsid proteins, and other enzymes, if required, are all packaged together to produce the complete viral particle. And the last stage is where the viruses actually are released by bursting the bacteria. So this is the productive process. Okay, this is the productive process. I'm trying. Okay. So for the attachment, as I mentioned, the adsorption step, the surface of the phage will have a specific molecule that recognizes a specific receptor on the host cell. In the case of the T4 phage, this protein is at the tip of these tail fibers, and it recognizes a specific molecule on the surface of the bacteria. Once the binding takes place, the virus will actually retract its tail fibers, and it will bury the tail spikes in the envelope of the bacteria by releasing an enzyme that can degrade the cell wall, T4 lysozyme. You know, lysozyme is an enzyme that can break down the peptidoglycan of the bacteria. So punching holes in the envelope of the bacteria, and then the virus will inject its genome into the bacteria. And from there, we have, of course, replication, synthesis of capsid proteins. The sequence of gene expression is very closely controlled. Well, it's inherently controlled by the actual genome. Okay. The early stages of infection after the release of the genome, there are specific proteins that are translated. These are referred to as early proteins. These include an enzyme that's called nuclease. And this nuclease will attack the E. coli DNA, in this case, the E. coli DNA, and degrade it. Then we have the genome synthesis, replication of the phage genome, and then we have late proteins are produced. And these the actual structural proteins that make up the capsid and the tail components. So when we have the assembly, is quite complex, in fact, and there are components that can self-assemble. So as soon as they're made, they will recognize their sort of partner protein and will bind to it. Others require protein scaffolds. So for it will be a large protein with extended sections, where different proteins can come together and bind to this scaffold. And then because they bind to the scaffold in close proximity, allow them to associate together, to assemble together. It can get quite complex, but quite intriguing, in fact. So after the viral progeny is completed, of course, the lysozyme produced in the latent infection digests the cell wall, causes the cell to burst, and the viral progeny released. That's all really for the productive infection or the virulent phage. So let's take another example for the other type, or the temperate phage of the infection. The best example that is still in use today, quite heavily, in fact, to look at genetics via bacterial and viral genetics, is the lambda phage. So this phage has a genome made up of double-stranded DNA, also infects E. coli. The genome itself is a linear piece of DNA, but the ends join together after injection into the E. coli to become circular. Now, infection of E. coli with this type of virus, and it can take on one of two forms: either the lytic infection, which we've looked at already. It will be the same type of infection as with the T4 phage. Okay. This particular virus can also go into the lysogenic infection, as shown here. So to the left in the blue arrows represent the direction of the typical lytic cycle. But depending on environmental conditions, this phage can actually go into the lysogenic infection, whereby it will integrate its own genome within the bacterial chromosome and remain like that for different periods of times. Okay, and as a result of this, of course, every time the E. coli, which at this point is referred to as the lysogen, every time the E. coli divides, it will divide along with it, of course, the genome of the phage. Okay. Now, when the bacteria encounters certain other environmental conditions, the virus can actually be induced. So this is a step called induction. So some examples could be damage to the DNA of the E. coli. It can induce the excision of the phage DNA and activation of the lytic cycle. Okay. When that happens, of course, and now the virus right away will synthesize the nuclease, which will degrade the E. coli DNA. Then it will multiply or replicate its own DNA, synthesize capsid proteins, and complete its lytic cycle. So the E. coli at this point is referred to as a lysogen. The rate of natural excision. So randomly during a division of a lysogen, we can expect, this has been determined experimentally, by the way, spontaneous excision of the viral DNA can happen in every 10,000 divisions. Okay? But we can experimentally accelerate this rate by exposing the lysogen to, for example, DNA damaging agents, whether it's chemical agents or ultraviolet. The viral genome appears to be able to sense this damage to the DNA of the bacteria. This would be, I guess, at the molecular level, translated into a sign of danger, and the virus decides to come out of the bacterial genome and replicate quickly before the damage can actually reach it. This is the process of phage induction. So, two important consequences to this form of bacterial infection. One is that the lysogen itself becomes resistant to superinfection. So, meaning that the lysogen does not allow infection by other phages. And the way it works is actually very clever. There is a protein that will not allow the viral genome to be excised and transcription to be activated. Okay, so a gene that controls the expression of other genes, or a protein that controls the expression of genes, is called the repressor. Okay? And it binds to, of course, upstream of those genes and does not allow for their transcription. Now, this repressor also happens to bind to the operator, which is the part of the DNA upstream of genes in the incoming viral or phage DNA, thereby preventing the gene expression of this other virus. So this is the immunity to superinfection. And the other phenomenon, or the other significant change, is the so-called lysogenic conversion, whereby the lysogen may actually show changes in phenotype due to the presence of the phage DNA, in which case, of course, we refer to it as prophage, right? So, and this is true also in animal viruses that are able to do the same. We have, you're familiar with the HIV virus, whereas genome can actually be integrated within the host, and it can remain there for years. Which means then some of the genes introduced by the phage can be translated, as is the case with the other bacterial genes. And these can include toxins. So we know that E. coli can exist in forms whereby it can release toxins, and other forms where it does not produce toxins. And the reason that it does not produce toxins is not because it's controlling the expression of the toxin genes, but it's rather due to the absence of the genes in its genome. So those toxin genes are brought into the bacteria by the phage genome itself. And in this case, we refer to this bacteria as lysogenic convert. The third type of phages, the filamentous phage, a very good example here, the so-called M13 phage, was discovered in Germany. It's a single-stranded DNA phage, that looks like long fibers, hence the name filamentous phage. Okay? It produces productive infections and not lysogenic infection. And this is the exception that I mentioned earlier. Here, the cells, infected cells actually do not die, but divide more slowly. See this missing text here from the publisher. Does not die, do not die, but divides more slowly than uninfected cells. The way this virus gains entry into the E. coli is quite intriguing. I have an image here. Oh yes, I'll take you directly to this image. Now you know that some bacteria is able to produce pili, right? Typically, it's a single pilus that is produced, and it connects two individual cells. In this case, this specific pilus is referred to as the F pilus. F stands for fertility. And this is a hollow tube through which the two bacteria can actually exchange genetic material, particularly plasmids that can be copied from the so-called F plus bacteria and sent through this hollow tube to the F minus bacteria. And then the F minus bacteria becomes F plus. Okay. And this genetic material, or this plasmid, now the recipient bacteria will have a copy of all the genes that are on the plasmid. These could include genes that will allow it to synthesize its own pilus. It can also be genes or plasmid that contains genes for antibiotic resistance, as I mentioned in a previous class. So the filamentous phage actually utilizes the F pilus to inject its DNA, following which it will take on the so-called replicative form, and then it will multiply in the normal case, in the normal process, replicate its genome, synthesize its capsid proteins. The capsid proteins are assembled through the membrane and the envelope of the bacteria. And then the genome is inserted into the structure, which is then released from the bacteria. Okay. So, the process of replication, of course, of the genome. Now, the bacteria has a single strand, in this case, sorry, the phage, has a single strand DNA virus, right? As mentioned here. So for it to be replicated, as you know, DNA replication, you've taken genetics before in biochemistry, you need a template from which the complementary strand is synthesized, right? So to be able to make copies of the single strand DNA, which let's call the positive strand or the sense strand, to make copy of like this one, we need to have a template from which we make copies. So to make the template, then the single strand DNA here, the positive sense itself, is used as a template to make the complementary strand, which is the antisense DNA strand. That's the replicative form of the genome. Then those two can be separated. The negative strand now is used as a template to make multiple copies of its own complementary strand, which would be the actual viral genome. Okay, just like a photocopier. And then the positive strand then is used actually for as to be packaged as an actual genome, right? This is the important point I've alluded to earlier, horizontal gene transfer. Now, bacteria is able to change its DNA. Well, it doesn't choose to change its DNA. Its DNA can be changed by a variety of ways, okay, one of which is the so-called transduction. And this is a consequence of phage infection of the bacteria, right? So now phage replication, the replication of the phage genome is not high fidelity, meaning that because of the speed and the frequency of multiplication, errors typically take place. Okay, this error can be in the actual replication of the genome, and it can also happen during phage assembly. Okay, now imagine, for example, during the process where the phage will produce the nuclease, which degrades the bacterial genome. During this process, the genome is fragmented into different sized fragments in the process. So some of these fragments can actually be included with the viral genome in the capsid during viral assembly. Okay. Now, when this virus, that's called transduction particle at this point, goes to infect another cell, it will take that DNA with it. Okay. So bacterial DNA in addition to phage DNA. And that DNA inside this new host can be integrated via homologous recombination, for example. That's a process you should be familiar with also from genetics, thus replacing the host DNA. So any gene essentially from a donor can be transferred to a recipient in this case. This is generalized transduction. Bacteria, in fact, can even take up fragments of DNA from the outside of the cell, from their environment. A process known as transformation. So I think here I may have, no, I don't have that. So this is schematically describes to you the process of horizontal gene transfer, right? Excision mistake during transition from lysogenic to lytic cycle. So in addition to packaging fragments of DNA during the degradation of the bacterial DNA itself, in the case of the lysogenic conversions, when the virus actually excises itself out after induction, it can take with it chunks of DNA from adjacent DNA sequences adjacent to its own genome from the bacterial genome during excision. And this would be packaged, of course, as part of the viral genome, but they're in fact bacterial. And the virus would then go on and infect another cell, thus transferring that DNA to this new bacteria. All right. That is how it goes. Okay, so let's go. Let us take a 5-minute break, and I'll come back and continue.
Dr. Saleh, I believe earlier you mentioned that I think it was viruses without capsids that are more resistant to cleaning agents. Would you be able to elaborate on that a little bit, please?
Yes, yes, of course, Alan. No, I mentioned not those without capsid, viruses without envelope.
Ah, yeah.
Those ones are more resistant than enveloped viruses.
Yes,
But-
non-enveloped viruses are more resistant to disinfectants. I just found that.
right.
Why?
Because the enveloped virus depends on its envelope for infection. Okay? Because that's where the surface molecules that will bind to the receptor on the host cell. Okay? Now, if you're treating those with disinfectants, particularly those containing alcohol, the envelope is removed along with the proteins that are within the envelope, which will allow the virus to bind to its host cell. And if those are absent, the virus is unable to actually infect the cell.
Makes sense.
The naked virus, on the other hand, does not have an envelope. It simply has a protein shell. Proteins are much more resistant, generally speaking, especially in the form of a capsid, okay? Because they form a shield that can be resistant to cleaning agents. So even alcohol is not able to actually remove the surface proteins, which are part of the capsid itself. And thus, the virus retains its ability to bind the host cell. Does that make sense?
Yeah, it does. I just didn't want to assume. I kind of guessed that, but wanted to make sure. Thank you.
All right, you're welcome. So I'm just trying to follow the chats here. Okay, I'm going on mute for a couple of minutes, okay? Okay. Now, so from this discussion now, you may have a better appreciation of the technical difficulty of cultivating viruses. Obviously, unlike bacteria, we cannot simply add media that we purchase, sterilize, and then inoculate with the virus, because it will not multiply. So we would need an actual host. In the case of bacteriophages, that's easier than other viruses, because bacteria is quite accessible. We have it, we can grow it in large numbers, and we can manipulate it in various ways. So for bacteriophages, to be able to cultivate them and to be able to count them, the size becomes another issue. Bacteria we can still count using the light microscope, but for the vast majority of viruses, we cannot do that. So we use a technique called the Plaque Assay. Okay? And this is where we exploit the fact that the virus, when it multiplies, it will kill the bacteria. It will be released. They will infect adjacent bacteria. They will lyse those adjacent bacteria, be released, and then infect adjacent ones, and so forth. So what this means is that the location where the phage was present on the lawn of a bacteria. So imagine you have a lawn of a bacteria on the surface of agar or within soft agar. Okay, so this cycle of bacterial infection, lysing, release, infection, lysing, release, will start to create a clear zone where there is no bacterial growth, because the bacterial cells have lysed. Okay, so this zone will increase in size as more and more phages are produced. Okay, this is what we call plaque. And this is how we count number of phages in a sample. Okay, we mix different dilutions of our sample that has the phage. Okay, we mix it with the bacteria, and then we apply it onto agar, and then incubate and wait for these clear zones to start to appear, those plaques. Now, the experimental assumption here is that if you are using the right dilution, that's why we typically do serial dilutions of the viral sample when we do this, these experiments. When you have the right dilution, you can safely assume that each plaque, if the plaque is uniform, round, and distant from other plaques, that's why you need to sort of do this before the size of the plaque gets too large and overlaps others, and also to use the right dilutions such that you have a low enough number of viral particles that are mixed in with the bacteria so that they don't crowd each other on the plate. If you control these parameters, you can safely assume that each plaque was derived from a single phage. Okay. So the number of plaques represent the number of phage particles in your sample, thereby providing you with a way to actually count the number of phages. Now, because we're not actually counting the particle itself, we are sort of indirectly counting it as a plaque. We refer to these as plaque forming units rather than actual phage particles. So when we want to identify a concentration of a phage in a vial, we identify it as PFUs per milliliter rather than viruses per milliliter. Okay. Is that clear? Yeah. Excellent. And this is the same, by the way, for animal viruses. Okay, so that's all for bacteriophages. Let's move on to animals and plants. So animal viruses, they share many of the features that we've looked at for bacterial viruses. Again, the virus, the steps are the same. You have the 5 stages: adsorption or attachment, genome entry, synthesis, assembly, and release. Okay. These steps are quite similar for the bacteriophages, except the attachment part and the genome entry. Okay. So the attachment part also involves recognition of surface proteins on the virus and receptors on the host cell. Okay. But the genome entry is quite different in the case of animal viruses. Okay. So let's take them one stage at a time. So for adsorption, again, the surface of the virus, if it happens to be a spiked virus, these proteins are actually on the tip of those spikes. So they will recognize a specific receptor in the host cell. It could be an integral glycoprotein or an integral protein without having any carbohydrates on it. Often there is actually more than a single receptor, in which case we would refer to the minor component as being a co-receptor. And this is true for the HIV virus, whereby it binds a CD molecule as a co-receptor. And if the cell does not have either of these those two receptors, then the infection rate is very, very low. Now, the specificity of infections of animal viruses, it's less stringent than that of bacterial viruses. We mentioned that an E. coli phage will not infect, say, Pseudomonas aeruginosa or Bacillus cereus or other bacteria. Now, because of the large number of this in complex organisms like ourselves, multicellular animals, we have many different types of cells, and we could have receptors for a specific virus on one type of cell, or we could have it on multiple types of cells in the same organism, right? We can also share homologous proteins, okay? So there are proteins on the surface of cells, say, of different animals that share significant sequence similarity and structural homology to our own, meaning that a virus that infects that animal may also infect us and vice versa, okay? So the nature of the virus, the specific type of surface molecule that it uses for adsorption, and the nature of the receptor to which the virus adsorbs will determine the host range. That's the right term, the host range for that specific viruses. Okay. So because of that, there are viruses that infect animals and not humans, viruses that infect humans and not animals, but there are those that can do both, simply because of the availability of the receptors in both that specific animal and humans. So rabies, we know, rabies can infect dogs and can infect humans, right? In other examples, the measles virus infects humans but does not infect dogs. Now, the next step, genome entry or injection into the host cell. This is where there is significant differences between bacteriophages and animal viruses. As you can see, the mechanism by which this takes place differs between naked and enveloped viruses. Okay, so in the case of enveloped viruses, they can release their genome in one of two ways. Okay, they can do it through fusion of their envelope. So remember, the envelope of the virus is essentially similar to a cytoplasmic membrane. Okay, it's a typical membrane or lipid bilayer, and it has membrane proteins and so forth. Now, we know one feature of membranes is that if they come close enough in contact with one another, they may fuse. So in the upper panel, as you can see, this fusion will essentially integrate the viral envelope with the cellular membrane of the host. And this causes the release of the capsid and then breakdown of the capsid to release the genome of the nucleic acid of the virus. So this is the fusion mechanism. The other mechanism is through endocytosis, a process you're also familiar with. Where it's a receptor-mediated endocytosis process. Binding to the receptor of the cell will cause the entire particle to be taken in through an endocytic vesicle. Then the virus will actually escape the vesicle, the endosome, then break down the capsid and release the genome in this case. And this would be the mechanism for the naked virus only. Remember, in fusion, of course, mechanism does not apply to naked viruses because they do not have an envelope. That's really the main difference between animal viruses and bacteriophages. And the rest of the steps are similar. You have synthesis, the synthesis step, where viral gene expression starts to take place to produce enzymes that are required for replication and to synthesize capsid proteins of the ribosome. And after that, of course, this process differs between bacteriophages and animal viruses in that some animal viruses, instead of an actual DNA for a genome, they have RNA for a genome. Okay, and this RNA can also be sense RNA or plus RNA or minus or antisense RNA genome. And I'll describe the difference shortly. Then there are those that are referred to as retroviruses. Retroviruses have RNA for a genome, but they are able to transform their genome into a DNA. And once, of course, it's in the form of DNA, it can be integrated within the host genome. Those viruses can integrate their genome within our chromosomes, and they can remain there for years. So a lot like the lysogenic cycle in bacteriophages. Okay, and these are called retroviruses simply because the fact that they actually go through the reverse process of RNA back to DNA. Okay, as you know, the normal process of processing of genetic material and proteins is that you start from DNA, then you go to mRNA, then you go to protein. Okay, going from RNA back to DNA is a reverse or retro in the retro process, such as the HIV virus. Okay. So let's look at the replication of the different forms of genomes, right? So double-stranded DNA viruses have, of course, both sense and antisense strands, right, of the DNA. The antisense one, the minus strand, can be used as a template to produce the messenger RNA, right? So this is the RNA form of the complement of the DNA, which is essentially the messenger RNA. Then that mRNA will be translated on the ribosome to a protein. Okay? The replication of the single-strand DNA viruses. So the complement strand has to be synthesized first, and then the complement is used as a template to produce more copies, as in a photocopier, to produce more copies of the viral genome. And those will be packaged eventually within the capsid. Okay. The minus strand will be used as a template to produce the messenger RNA in this case. Right. So here we go, double-stranded DNA. The minus strand is used for transcription to make the positive strand RNA, positive single-strand RNA, which is the messenger RNA. Translation on the ribosome, proteins. The minus strand then is replicated, a complement copy is made, and the complement of the press strand is made to produce the double-stranded DNA. For the single strand, a complement is made here in the blue. And once you have a double-stranded DNA, then it's the same sequence as in the double-stranded DNA virus. Okay. The negative strand or the antisense strand is used to transcribe messenger RNAs or protein synthesis. And the other one is used to synthesize the positive strand, which is essentially the actual genome of the virus. This step, DNA synthesis of the DNA viral genome takes place in the nucleus, right? RNA viruses are replicated in the cytoplasm, okay, because they don't need any of the DNA enzymes that are typically found in the nucleus, right? One key enzyme that is required in this case, which is encoded by the viral genome itself, is an RNA-dependent RNA polymerase, okay, replicase. So this is an enzyme that is actually capable of making complementary strands of RNA from a template that is an actual RNA, not a DNA. Okay. This strategy, of course, obviously varies with the viral genome. The single-strand RNA could be a positive or sense strand, just like the DNA, or it could be an antisense or negative stranded RNA genome. The positive sense, the positive strand or the sense strand is equivalent to a messenger RNA and can be directly translated on the ribosome to make proteins. Okay. So the strategy here, if we look at the single strand positive sense RNA, I shouldn't say positive sense, either positive strand or sense RNA. So it can go directly, as I mentioned, directly onto the ribosome for translation because it's equivalent to mRNA. Okay. Now for genome synthesis, you need to make the complementary strand using the RNA-dependent RNA polymerase. Then this negative sense RNA is used as a template to make multiple copies of the complement, which is the positive sense strand, which is the actual viral genome. And then these are packaged within the capsid. Okay, if it's an antisense RNA genome, first the complementary strand is made again with an RNA-dependent RNA polymerase to make a positive sense RNA. This can act as mRNA for translation on the ribosome to protein, or it can be used to make the complementary strand multiple copies of the complementary strand, which is essentially the viral genome that is packaged within the capsid. And if it is a double-stranded RNA genome, which does exist, okay, we have first synthesis of a single strand from the negative strand of the RNA. This positive sense or mRNA can be translated to produce capsid proteins and other enzymes required for the completion of the cycle, or it can be used to synthesize the complementary strand to create the double-stranded RNA genome, which is the actual viral genome that is packaged within the capsid. Okay, I hope that is clear. Right. So now replication of RNA viruses. Then these viruses create problems for us because of their poor proofreading ability. Okay, poor, actually I should say lack. Proofreading, again, you should recognize this term from genetics, right? Enzymes that replicate nucleic acids, particularly DNA, right? DNA polymerase. It has proofreading ability, meaning that every time it synthesizes few complementary nucleotides from a template, it will go back and then sort of slide along those nucleotides that it has synthesized to make sure they are correct. They are in fact complementary nucleotides to the nucleotides in the template, right? So that's called proofreading. Okay, and now viral replicases lack this ability, which means that they generate a much higher rate of mutations during replication step. And this obviously results in what we call antigenic drift. Yeah. So viruses, that's why these RNA viruses hypermutate. Okay. Every time one cycle of viral replication is completed, some of these viruses are in fact different than the original ones, and their percentage within the viral progeny increases as they infect adjacent cells, multiply themselves again, and then that produces additional mutations, and these are called antigenic drifts. There is another process that can produce mutations. It's called antigenic shift. In the antigenic shift happens when the virus that is infecting the cell contains genome that is segmented rather than a single linear RNA or DNA genome. It's a segmented genome. So here, unlike the case of bacteriophages, where once a bacterium is infected with a phage, it becomes immune to infection with other phages, as we mentioned. In this case, actually, this does not happen, unfortunately, for us. And multiple viruses can infect the same host, or the same cell. Okay? And their genomes, therefore, once they are released and they're replicated, different fragments of different viruses can then be packaged within the same capsid, of course, producing now different viruses. So this is a large-scale mutation that we call antigenic shift or chromosome reassortment. Right. So now let's go to the retroviruses, as we mentioned earlier. such as the HIV virus. So these are carry an enzyme called reverse transcriptase. Again, the name is self-explanatory. It is able to make DNA from an RNA template. So in the case of the HIV, its genome is a sense strand RNA genome. The reverse transcriptase that is carried typically with the virus will synthesize the DNA strand. Then a complementary strand is synthesized from the host using host enzymes that produces a double-stranded DNA genome of the virus. Then this can be integrated, in fact, into the host chromosome and remain inactive within the host for many years. Or it can go into immediately into an active stage where the virus is used, of course, to transcribe messenger RNA to make capsid proteins and other enzymes that the virus will need, and then replicate the genome, which is essentially the entire mRNA of the genome. That's the actual genome of the virus. The assembly and release are similar to those of bacteriophages, except the release step, where for most bacteriophages, as we mentioned, it involves the lysis of the bacterial cell, except for the filamentous phage. And the same for animal viruses. Some viruses are released by actually lysing and killing the host cell, and some are released slowly while the cell remains alive, or it becomes more like a zombie, if you will. The assembly process is similar again, although some animal viruses may be more complex and involve more steps, but essentially the same. Some components self-assemble, and the nucleic acid or the genome of the virus will recognize the self-assembled template of the capsid, and they all become associated together, and then migrate towards this step. Actually may take place within the membrane of the cell itself. In the case of enveloped viruses, as the assembly is complete, the virus is already at the membrane of the cell, then it will excise itself out of the cell, taking with it that membrane which originated from the host cell. That's in the case of enveloped viruses. In the case of naked viruses, more often than in the case of enveloped viruses, naked viruses will tend to lyse the cell for release. Okay. So this is schematically the release step for the enveloped virus. Now, as you can see, here we have a complete nucleocapsid. Some of the viral proteins are actually synthesized within the endoplasmic reticulum, and from there they're exported all the way to the membrane. That's why it depends on the virus, obviously. Okay, but infected cells, our own cells, will display on their membrane viral components. So we can, with antibodies, identify cells that have the virus inside, if microscopy is not possible in this case. Then the capsid will recognize these viral proteins in the membrane of the cell. It will associate with it, then it will excise itself, taking with it the cell membrane, which becomes the envelope of the virus. Okay, so the viral envelope contains virus-specific proteins. It can also contain host-specific proteins that are stripped away with the membrane during its release. So in the case of animal viruses, so we have the acute infection, which is equivalent to the lytic cycle in bacteriophages, okay, where the virus right away starts multiplying and then it is released. So this is a rapid onset of the infection, short duration, release of the virus. That's where the individual shows actual highest symptoms of the illness. And the immune system gradually over this time develops immunity in most cases. And we find these acute infections in viral infections such as the influenza, mumps, and the poliomyelitis, and the poliovirus. But there are those that produce persistent infections. So these are infections that last for years, and they are of two forms. Okay, there are the persistent chronic infections and the persistent latent infections. The persistent chronic infections is where the virus is active throughout this period, but it's active at a low level. Okay, so the virus is multiplying slowly. There is no clinical symptoms of disease, and the person would actually be shedding viruses. Okay, it could be, of course, transmissible at that point. In the case of the latent infection, the viral genome is actually silent. So this is like the lysogen in the case of bacteriophages. And this again can be induced. So there could be an induction at some point due to changes in physiology of the host, can activate the virus, and it will go into the actual active infection. Okay. The virus, again, within the chromosome, in the inactive form, it's referred to as provirus. In the case of bacteriophages, it's referred to as prophage, right? So if we look at these different types of infections and look at clinical symptoms, okay, schematically, the top view in panel A shows the increase in clinical symptoms over time. So you have sharp increase, lasts for a short time, then decrease of clinical symptoms. This is in the case of an acute infection. In the case of a chronic infection, you have the clinical symptoms actually increasing in size rapidly, staying for a short period of time as actively multiplying, and then it does not disappear. It stays for extended periods of time, where the virus is at a low rate, is being divided, is being replicated and released at a slow rate without a change in the level of clinical symptoms. Okay, they remain steady, and these might be mild. In the case of the latent infection, as you can see, you have the initial infection normally. As an example here is cold sores, the virus that causes the herpes virus that causes cold sores. Typical infection, multiplication, so maximal level of clinical symptoms, then decrease in the number of viral particles being produced, then it remains silent for extended periods of time, where of course the host at this point is non-infectious. So unlike the chronic infection, at some point down the line, of course, there's activation of the virus, then it will go through another active stage of infection, and it produces clinical symptoms. Then back down to a silent infection again. And this cycle can be repeated, of course, over and over. Now, because of what we mentioned about the ability of viruses for mutations, their ability to introduce new DNA into the host, and their ability to integrate their chromosome into our own DNA. This will alter, obviously, the balance or physiological balance of our cells. And this would be the sort of simplest definition of cancer, right? It's an imbalance in cell physiology and loss of control of cell replication, right? So there are two types of genes in our cells. Those that are called proto-oncogenes, and these typically stimulate cell growth and division. And there are those that balance the activity of these genes that are called tumor suppressor genes that inhibit cell growth. Okay. These are typically at a balance. Okay. Mutations in these, which can happen by the virus bringing in, say, copies of other oncogenes, or when integrating into the chromosome, affecting the expression of the proto-oncogenes or the tumor suppressor genes. Okay. If you inactivate a tumor suppressor gene, it means the cell lost its ability to inhibit the growth. Okay, so any imbalance produced as a result of the viral infection in these particular gene classes can essentially cause the cell to lose control of its growth, right? So the genes that are called proto-oncogenes are genes that, if mutated, can be transformed onto an oncogene. An oncogene is, of course, a gene that can cause uncontrolled cell growth. That's why the field of cancer in medicine is referred to as oncology, right? So again, so viruses can carry oncogenes, and during infection, these genes are active and promote the cell to grow. Or if the virus integrates its genome within the chromosome, the site of integration could interrupt tumor suppressor genes, so that now the cell is unable to actually balance its growth signals by counteracting that with suppressing other genes, and thereby causing eventually cancer. So viruses that can cause cancer are referred to as oncoviruses. And most virus-induced tumors are associated with DNA viruses, although some are RNA viruses, in fact. Now, a few things to keep in mind when we talk about oncoviruses and cancers that they can cause is that cancers do not develop immediately following viral infection. In fact, take many, many years, decades, for this to appear. Some small percentage of those infected with those viruses still even do not develop cancers 15 to 40 years after infection. So the infection with the oncovirus does not mean automatically cancer will appear 15 to 40 years after that infection. Today, we have actually developed effective vaccines against some of these, like the human papillomavirus. So here are viruses associated with cancers in humans. There's 7 of them. The type of nucleic acid it has for genome, and the type of tumor that they can cause. The Epstein-Barr virus, hepatitis B virus, herpes virus type 8, human papillomavirus, Merkel cell polyoma virus, hepatitis C, and the last one, should correct here, I have it in the red. The publisher have this, the name incorrect. It should be HTLV1. That's a human T lymphoma virus type 1. So HTLV1, not HTVL1. Okay. So looking at the overall effects of animal viruses on the cells that they infect. So viral DNA integrates into the host, can cause tumor, or it can remain silent and cause a latent infection. Here we have viral DNA causing or forming a plasmid, so not integrating into the chromosome. It can still cause tumors because it would carry genes that are potentially called oncogenes, which stimulate the cell growth, uncontrolled cell growth, or it can remain in a latent infection, or the virus immediately goes into the replicative cycle, replicate, and then the result of the replication, either the cell is killed, it bursts open, releases the viral progeny, or we have slow chronic infection where the virus is slowly released from the cell while the cell remains alive. Okay, through the process of budding. More recently, or in the past 10 to 15 years, scientists have actually been trying to develop oncolytic viruses, or viruses that can be targeted to tumors. Oh, I better get my charger. My battery is going down. Hang in there. Okay, so viruses that can be targeted to tumors, and then those viruses will infect the tumor cells, multiply, and end up destroying the cancer cells, right? So these are genetically engineered viruses. First, of course, you need to try and target the virus to cancers, to the specific tumor cells. And some cancer cells actually can be differentiated from normal cells, from healthy cells, by expressing, by having larger or overexpression of certain surface receptors. So those can be exploited to target tumors at a higher percentage, of course, than healthy cells, because you need some type of specificity, right? In targeting the cells with those viruses. And then once you accomplish that, the virus will automatically penetrate the tumor cells, multiply, and end up killing the tumor. And the first, in fact, oncolytic virus that was approved by FDA in the United States was back in 2015, so quite recent. And this was based on herpes simplex virus type 1 to treat advanced inoperable melanoma tumors. In terms of cultivating and quantifying animal viruses, so here we have the same issue with bacteriophages. We need to supply appropriate host for the virus. Okay? In the case of the bacteriophages, it's easy. Bacteria, E Coli, we can grow it. It multiplies quite rapidly. doubling rate in the range of 20 minutes. It's easy to grow. For animal viruses, it's not that straightforward. In the old days, they used to actually cultivate the virus by infecting the entire animal and then recovering the virus from that host. They've also used to inoculate fertilized chicken eggs, and this is still in practice today. Vaccines, viral vaccines, require, of course, large numbers of viral particles that have been inactivated, right? So to generate these large numbers of viral particles, they inoculate fertilized chicken eggs, then incubate them, and then recover the virus from those eggs after incubation, and inactivating the virus, say via chemical treatments. And these can be used directly in a vaccine after that. Then, of course, with the development of tissue culture, we are able to actually separate cells from different tissues. We can culture those cells, and then we can use them as hosts for a specific virus, right? It depends on the virus. It will limit really the selection of cells that we can use. Nonetheless, it's a much cheaper and easier process to do. When we started working with these cells as hosts to cultivate viruses, we've also noticed that viral infection causes some microscopically visible cytopathic effects. So cells may change their shape as a result of the infection. They can detach from the surface of the plastic in the tissue culture flask. They can lyse. They can fuse into giant cells called syncytium. Or the virus can actually collect within the cell and form dense, microscopically visible, dense regions within the cell that we call inclusion bodies. You're familiar with. In terms of quantifying viruses, it's a similar way to bacteriophages. Okay, we use the Plaque Assay. We can culture the host cell until they form a complete layer, like a lawn, and then inoculate with different serial dilutions of our virus sample, and then wait for clear zones to show, to become visible, and then we can count these as plaques. Okay, there's also the Quantal Assay, whereby the serial dilutions of the virus are given to hosts, okay, an actual animal, okay? And you can, from the result of the infection, you can either recover the so-called infective dose, that is the dose that causes infection in 50% of the infected animals, or you can extract the number for the lethal dose, the dose that kills 50% of the hosts in the case of experimental animals. In some viruses, not all of them, in some viruses, there is actually an easier, cheaper, and faster way to do this. Some viruses are able to agglutinate red blood cells because they have surface proteins that can bind receptors on red blood cells. Okay, because of the geometry of the virus will have copies of that surface protein on different sides of the capsid, and therefore can associate more than a single red blood cell on its surface. And as the numbers increase, as you can see on the right there, these eventually will grow in size to form clumps of viruses and cells together. Okay, so this process is referred to as agglutination, And here again, the publisher made a mistake. You can correct this in your slides. It says mixed virus with dilutions of red blood cells, but no, it's actually you mix red blood cells with dilutions of the virus. And if you have the right proportion of number of viruses and the number of red blood cells such that you can successfully form this large macroscopically visible, so you can judge this reaction with your eyes, in fact. If you have the right proportion of these two together, you will form these large visible aggregates, and these are visible if you do it in these sort of kind of test. If you could see here, you have different dilutions of the virus mixed with the red blood cells. If there is no agglutination, the red blood cells in few minutes they settle to the bottom. Okay? If there's agglutination, this large crystal lattice will form, and you will actually you will be able to see the difference between this form as shown here as complete red from a negative reaction where there's no agglutination, where the red blood cells simply settle to the bottom of these wells. So the highest dilution with maximum agglutination is referred to as the titer of the virus. Okay, so in this case, this is the highest dilution, 1 in 256. So the titer of this particular virus, strain A, the titer is 256. Okay. For viral B, it's 32. For C, it's 512, and so on. That's called the titer. Plant viruses, they of course they are of economic importance because they can destroy the crops. And plants that are infected with viruses, just like in animals, they produce symptoms. Visible signs we see in plants: yellowing of the foliage, irregular lines on the leaves and the fruit. Individual cells or organs may die. You can actually see a change in the plant itself. Tumors may also appear because, again, plant cells can also suffer from lack of balance of cell regulation, and uncontrolled cell growth can be produced by certain viral infections. Plants can also show stunted growth, very small, short plants. So it can also produce deformation of structures. Okay, you may have noticed in some trees there are bulges in the trunks of the plant that looks odd. These are likely produced by viral infections. So plants generally do not recover from viral infections because they don't have an actual immune system like we do. Okay, and viruses actually gain access to the plant and infect the plant in a different way. So they don't depend on adsorption to the surface of the plant cell, adsorption to a receptor on the plant cell, but rather they gain entry into the plant as a result of injury to the trunk or the leaves or the stem of the plant. And then from there, they will actually start to multiply and cause damage to the plant. The tobacco mosaic virus, it's very difficult to eliminate because it multiplies to large numbers. And in fact, if you were to take an infected tobacco plant, up to 10% of the weight, the dry weight of the plant is the actual virus. That's how abundant it is in the tobacco plants. So these are some signs. You can see lines on the leaves, yellowing spots on the leaves as well, depending on the plant itself. And they are quite resistant to environmental conditions, such as high temperatures, low temperatures, dryness. So they're quite problematic to eliminate. Okay, now we move to something even more exciting. But before so, let's take another quick five. Okay. We're both back. Okay, so we talked about very small viruses. We talked about even smaller viruses that are able to parasitize other viruses, as in the case of Sputnik and the Mimi virus. Now, I don't like to use the term a virus infects another virus, okay, because this is not how it actually happens. Sputnik does not actually invade and infect the Mimi virus. Okay, it parasitizes it during co-infection, and in fact, there are even bacterial examples of this. If you look up the delta agent in the case of hepatitis, you'll find that this hepatitis D virus actually only multiplies when it's co-infecting the same cell with another hepatitis virus. Here we're going to talk about entities that are even smaller than the smallest viruses. These are, in fact, not even a particle, they're single molecules. Okay, the first we'll talk about RNA molecules, and then we'll talk about proteins, a protein molecule. The RNA molecule here is called viroid. It's a single-strand piece of RNA molecule with a closed ring, and this can be synthesized actually fairly easily in the laboratory. These are only found in plants. They infect plants. They can cause serious disease, including the potato spindle, tuber, chrysanthemum, stunt, citrus, exocortis, cucumber pale fruit, and kadang, which essentially infects pineapple plants and palm trees, certain species of palm trees. So just like plant viruses, they enter through wounds in the leaves or the stems, and once they penetrate the plant body itself, they can multiply, and from there they can be released, and they can infect other plants. Imagine a single molecule of RNA being able to do that, and this may take you back in the theory of evolution to the ability of simple molecules to come together, like amino acids or nucleotides, to form a single molecule that is in a way able to carry enzymatic activity with it, or allow itself to multiply in other hosts. For the proteins, one you're familiar with, that is the causative agent of a number of related diseases, mad cow disease, the sheep and goat scrapies disease, and the chronic wasting disease in elk and deer. In humans, they cause the so-called Creutzfeldt-Jakob disease and the fatal familial insomnia, as well as Kuru. Kuru is in fact the first human disease discovered to be caused by this unknown entity at the time, and this was essentially resulted from a religious practice where brain tissue of deceased relatives is spread on the extremities and the arms and the hands of certain relatives, including some children, which caused obviously the disease to spread, because this protein is in fact an infectious agent of the central nervous system. It attacks the brain. In our brain tissue, so here we have in panel A normal tissue, and in panel B you have an infected tissue, and you can see these clear zones that are produced as a result of infection with the prion protein. And these clear zones, tissue look like sponge. Hence, the general term that is used to describe all prion diseases, spongiform encephalopathies. Encephalopathies, of course, are diseases of the brain. And the fact that it is transmissible, therefore the name becomes transmissible spongiform encephalopathies for all of these prion diseases. Okay. And the clear zones, of course, appear as a result of neurons that end up dying, of course, and then they are degraded and produces these clear zones. And eventually, as the number and size of these zones increase, the brain deteriorates over time. Eventually, the infected individual succumbs to the disease. Now, in our brain tissue, we have the normal form of the prion protein. It's called PRPC. Okay. This is typically a membrane protein that has been shown to be implicated in copper transport and other activities. But when the tissue is exposed or is infected with a mutant form of this protein that is referred to as PRP superscript as SC. Okay, superscript SC. What happens is that this is how the initial questions when the disease was discovered is how can a single protein actually multiply? Because unlike RNA and DNA, there are no enzymes that can actually make copies of a protein, right? It would be great if we discover some, or we're able to actually invent such a process, but it does not exist. So the question was, how does this protein actually multiplies? And of course, the discovery that shed light on this process was the finding that this mutant protein shown in red, when present in the brain after infection, it will associate with the normal cellular form of that protein, transforming it into a mutant form. Then those will then associate with other normal proteins and transform them into mutant forms. And the mutant form has different biophysical properties, including its ability to aggregate. And these form aggregates that can also increase in size, cause neural death, and produce visible plaques, microscopically visible plaques, a lot like the amyloid plaques that we see in Alzheimer's disease, because amyloid plaques are essentially aggregates of peptides as well. So a very similar process. And that's how this protein is able to actually artificially multiply. It does not multiply by replication, but by increasing the number of mutant forms of the normal protein. And that, ladies and gentlemen, brings us to the conclusion of this chapter. Are you still with me?
Yep.
Excellent, excellent. Any questions you have on this material, or anything related or unrelated?
I have a question.
Yeah, go ahead.
I was wondering, do we know what the origin of viruses come from?
Not quite, but you can think of them as remnants. I mean, that is my personal view, is that they are residuals of the production of the primitive cells, when fragments of DNA and RNA were being released, that eventually were contained within proteins that are able to adsorb to other living entities like bacteria and eukaryotic cells. And from that point on, they of course became part of life on the planet. Now, if you're asking about the actual, the single ancestor of all viruses, I don't think they exist, or we don't know yet of a single ancestor of viruses. That's the short answer.
Okay, thank you.
You're welcome. Any others? If there are no other questions, by the way, we're done. So you can, those of you who want to leave, you can leave. Thank you, everyone.
I had one more question real quickly.
Go ahead.
So could viruses develop in a place where there's no life forms?
I don't know. That's a tricky question, because remember, for the virus to occur, as I mentioned earlier in expressing my personal view, is that it's a residual process from the actual existence of an actual living cell, whereby genetic material lost or fragmented living cell released this genetic material that eventually encapsulated within a protein and became a virus. So for a virus to exist in the absence of these living cells, if it does happen, it could happen. I can see it as a possibility, but it would not survive, obviously, because there are no living organisms for it to infect and multiply. So
could it just be dormant until like one day when living organisms do come?
It's possible, absolutely, if the conditions, if the basic criteria are met. And remember, the basic criteria is that the virus comes in contact with an entity that is able to adsorb to and penetrate. If those criteria are met, then it is quite possible, absolutely. It is quite possible. So let's take this hypothetical situation to another level. Let's say, is it possible that a meteorite will arrive on the planet and will carry within it a cosmic virus, right? An entity, it's possible that a biological entity such as a virus, as what we recognize as a virus, can come from space. Whether or not it will be able to actually have proteins that are able to recognize receptors on living organisms on our planet would be another question. Because that virus would have developed on a different distant planet, for example. It would have developed away from the biological entities that are on Earth. Therefore, the probability of it being able to find a receptor on cells of organisms on Earth would be quite remote, in my view.
Okay,
Yet
so
a lot of spaceships are constructed in almost sterile environment to prevent any microorganisms and viruses to be on the ships.
Yes, well, yes, Helen. You know, it's a hypothetical situation where, let's say, life started at a distant planet, and then that planet exploded, and then fragments were flowing in space, and then one of them crashed on Earth. It's not as a remote possibility as the possibility of that virus eventually on Earth being able to actually infect cells on Earth and multiply. I think that's even more remote than the virus developing on a distant planet.
okay, so overall, it is possible, but the chances of it, one, happening, and two, being able to affect receptors is so small. It's basically not possible.
Yes. I wouldn't use not possible. I'll say quite very remote. Very remote. I can't put a number on that probability, But I'd say very remote. Is it impossible? I can't say with absolute certainty that it cannot happen. No, I cannot say that. But it would.
Based non-sequential, however, it's so
Yes.
small that it doesn't bear worrying about, basically.
Yeah, I would agree with that statement.
Okay, thank you. That was very interesting.
You're welcome.
Have a great week, and I'll see you next week.
You too, everyone. Take care, be safe, and we'll chat again next week. Have a good one.
Have a good day.
Bye-bye.